We're thankful that she rose to the occasion today to be our next student speaker. So please help me in welcoming, with the title, Mapping Rare Specific Black Spot Disease Resistance in Tetraploid Landscape Roses, Cindy Rouet.
Good morning, everyone. Thank you so much for the introduction. So, as mentioned, my name is Cindy Rouet, and I am a PhD student at the University of Guelph. I execute my research in rose breeding at Vineland Research and Innovation Station, which is a non-for-profit organization that's dedicated to innovation in horticulture, and that's really applied research. So my mission is to modernize Canada's national rose breeding program by implementing molecular marker assist selection. And my presentation today is going to focus on mapping black spot disease resistance. So I don't know how many of you are familiar with roses, so I would like to quickly introduce the history of roses. The domestication of roses has been long and complex with a rich historical background. So the domestication of roses has been long and complex. The rose is native from different temperate regions from the northern hemisphere, and the wild rose has been known in China for over 5,000 years. We also know that the Western world is a center of diversity for roses. The Chinese rose was brought to Europe early 18th centuries, and the first hybridization between Chinese roses, European roses, and Middle East roses gave birth to the Portland roses and the hybrid perpetual, just to cite a few. The contribution of Chinese roses was major, with the recurrent flowering trait. The introduction of the first hybrid La France in 1867 marked the beginning of a new class of rose, the modern roses. That include the hybrid tea, famous Polyantha, and Floribunda, that are characterized by increased level of hardiness, disease resistance, wider range of colors, and bigger flowers. So to date, we count about 200 rose species, but we know that only 10 to 15 contributed to the genetic background of the modern germplasm. So roses have been used for multiple uses, for the production of rose essential oil, for cooking, in medicine, for cut flowers, and also to beautify our landscapes. So the rose breeding program at Vineland takes about 10 to 13 years to release a rose. So between the initial cross to the release on the Canadian, US, or European market. So far, breeding has been achieved through conventional breeding technique only, which slows down the release of new varieties of roses. So in most breeding programs, roses are selected based on the breeder preferences. One of the unique features of Vineland is that everything from the selection to the marketing of a rose is guided by consumers' insight. We have a trained sensory panel and a consumer panel, and we are aiming to release roses with this whole package that meets all consumers' expectations in terms of color and care. So cold hardiness and black spot disease resistance are our major targets in our rose breeding program to release high quality and low maintenance roses. So cold hardiness and black spot disease resistance for us are good targets to develop marker assist selection to speed up our program and release roses in a more accurate way. So when we are working with roses, we have the luxury to work with a small genome of only 500 megabase pair. However, even if the genome is small, because the domestication of roses results of centuries of hybridization events, polyploidization, and incomplete speciation, most modern roses are tetraploid, which complicates our genetic studies. So I would like to now focus on mapping black spot disease resistance. So black spot disease is one of the most important foliar diseases of roses. It's caused by a lascomycete fungus obligate to the genus rosae that overwinters in the fallen leaves and infected canes and spread through splashing. It's really difficult to breed for black spot disease resistance using a visual rating from the field. And the fact that several races of black spot exist in our field is really challenging for the rose breeder. So now I would like to focus on how we map black spot. So in order to map black spot disease resistance, we used a segregating mapping population between a resistant rose genotype 60, that is a key parent in our breeding, and a susceptible cultivar singing in the rain. We used 3 different isolates of Diplocarpon rosae, including 2 that are representative of the diversity that we have in our experimental field. And we used an improved detached assay that consists of inoculating a detached rose leaf with single spore inoculum. And after 2 weeks of incubation, we rate our lesions on a scale from 0 to 5 based on the presence of Acervali and Conidia. So what we observed is that for each of the 3 isolates tested in vitro, the segregation between the resistant phenotypes and the susceptible phenotypes was binomial. And we observed the same segregation pattern for data that was collected in the field under natural infection, which suggests that we have a gene-for-gene type of interaction. In addition, we know from literature that black spot disease is controlled by a gene-for-gene type of interaction, and we have so far 4 genes that have been identified, and we know them from RDR1 to RDR4. So we generated genetic maps for both the male and the female of the cross using genotyping by sequencing, following a 2-way pseudotest cross strategy in a tetraploid cross, using only markers that are in simplex configuration. Also, we used the world reference genome sequence of rosae chinensis in order to estimate the physical position of our markers in our linkage maps and identified the 7 linkage groups. So we used the female map that we saw on the picture before in order to perform a QTL analysis on the data collected in vitro and the data collected from the field. So we performed interval mapping and multiple QTL mapping, and what came out of our QTL analysis is that the resistance to the 3 isolates tested in vitro and field resistance collocated at the telomeric end of linkage group 1. Also, we can see that the QTL explain a really high percentage of the variance observed. So we know from literature that the resistant locus RDR1 is located on the telomeric end of chromosome 1 in the rose genome. So RDR1 locus is coming from rosae multiflora. It's a cluster of R gene that confers broad spectrum resistance to black spot disease. So what we did, we actually performed an alignment between the publicly available sequence of the mu-RDR1 locus and the whole sequence of rosae chinensis on the left. And we observed that the mu-RDR1 locus align at the telomeric end of chromosome 1 in rosae chinensis. So because we used rosae chinensis in order to estimate the physical position of our markers, by comparison, we were able to see that our QTL were physically located in the RDR1 locus. So the next step, we know from functional genomic studies that within this RDR1 locus, the mu-RDR1A gene is the active gene within this locus that confers broad spectrum resistance to black spot. So the sequence of this gene is available, and we actually align the sequence of both parents of the cross with this public available of the active genes of the RDR1 locus. And we observe that the susceptible parent has a deletion in this sequence, and that the unique sequence present in the resistant genotype 60 is involved in the resistance. So we developed two pairs of primers based on this unique sequence in the resistance parents to develop two types of markers. We developed two diagnostic markers, one presence absence and one indel marker. So we validated those two diagnostic markers using high-resolution melting technology that allows us to detect the presence of the resistant allele using the shape of the melting curves. So we used two validation populations to validate our markers. We used the mapping population, 60 into Singing in the Rain, and we used a test cross population between the genotype 60 and the genotype S1310 that are both intern to our program. And S1310 is derivative from 60. So for the presence absence marker, we can clearly identify the progeny that carries the resistant allele that's inherited from 60. And for the indel marker that's codominant, we can definitely identify the heterozygous genotypes, but in the case of the test cross population, we can also estimate the dosage of the resistant allele. So this marker data align pretty well with the phenotypic data. So those are the phenotypic data for the mapping population collected in vitro and collected in the field. We have a few misclassification for two of the isolates in terms of progeny not having the resistant allele, but being resistant in the detached assay. And this could be due to the difficult nature of the detached assay itself. So those markers for us will be of major contribution. I screened a large collection of germplasm of about 600 genotypes, and we observed a really good alignment between the presence of the resistant allele and field resistance. So those markers are going to be used to screen our progeny at early stage of the breeding program to select for black spot disease resistance. So those markers are highly specific to the genotype 60 because they've been designed based on the unique sequence in this genotype. 60 is a key founder of our program, and we have multiple derivatives of these genotypes that constitute, that are key in our collection of parental germplasm. So now we're able to develop markers that are associated with black spot disease resistance. We want to develop markers associated with cold hardiness using artificial method and field data. So to conclude, I would like to mention that using GBS technology and HRM, we're able to develop markers that are associated with cold hardiness and disease resistance. We are going to use this knowledge to develop more markers associated with traits of major interest, such as fragrance and growth habits. And we are using this technology to release, in a quick way, low maintenance and high quality roses, improving the competitiveness of canine nurseries. And Vineland is releasing roses under its 49th parallel collection. So our first rose was Canine Shield, and we have two roses coming up soon, including Chinook Sunrise coming up this summer, and Aurora Borealis coming up next year. So thank you so much for your attention. I would like to thank everyone from the organizing committee to have me here today. That's such an honor. Thank you so much, and I will take any question.